I'm Semenkovich from Washington University in St. Louis, and I'm going to tell you about our recent paper in Cell Metabolism dealing with diabetes. The loss of functional beta cells causes diabetes. Guifang Dong, Wei, other colleagues, and I discovered that a post-translational modification is involved in beta cell dysfunction in type 2 diabetes. This process is called palmitoylation, and it involves formation of a bond between the common fatty acid palmitate and a cysteine residue. Many human proteins are palmitoylated, which affects protein trafficking mostly by altering membrane affinity. Unlike other lipid modifications such as myristylation and farnesylation, S-palmitoylation is reversible. A large family of enzymes called acyl-transferases add palmitate to proteins, and a small family of enzymes remove palmitate from proteins. This sequential addition of a lipid mark, followed by erasing that lipid mark, is dynamic and necessary for rapid membrane signaling events. Most depalmitoylation, erasing the lipid mark, is carried out by the enzyme acyl-protein thioesterase 1, or APT1 for short. People with type 2 diabetes usually have a combination of insulin resistance and insulin hypersecretion, but several studies suggest that primary insulin hypersecretion, independent of insulin resistance, predicts developing type 2 diabetes. We found that culturing normal human pancreatic islets under hyperglycemic conditions decreased activity of APT1, the enzyme that removes the palmitate mark from proteins. APT1 enzyme activity was decreased in islets from people with type 2 diabetes, and APT1 knockdown in healthy human islets caused increased insulin secretion. Based on this information, we generated global APT1 knockout mice and found they had improved glucose tolerance and increased insulin levels in glucose tolerance tests. APT1 deficiency did not affect insulin sensitivity or body weight. The increased insulin secretion was islet cell autonomous and was unrelated to calcium influx or insulin biosynthesis. Instead, increased insulin secretion was associated with prolonged insulin granule fusion. We identified SCAMP1, a vesicle protein known to mediate exocytosis in other cell types, as an APT1 substrate in islets. SCAMP1 knockdown increased glucose-stimulated insulin secretion, and insulin hypersecretion and APT1 deficiency was reversed by an engineered SCAMP1 protein. We also generated islet-specific APT1 knockout mice, and these animals also showed insulin hypersecretion with normal insulin sensitivity. One might expect APT1 deficient mice with insulin hypersecretion to be protected from diabetes, but we found the opposite. APT1 deficient mice fed a high-fat diet, and APT1 knockouts in the DB-DB model showed glucose intolerance, decreased insulin levels, and beta cell loss compared to controls. Obesity promotes diabetes and is associated with increased tissue stores of palmitate. Our findings suggest that altered palmitoylation of key proteins mediating exocytosis could promote beta cell failure, and that correcting this process might preserve beta cell function and prevent or delay some forms of diabetes. Thanks for listening.